Welcome everyone to BrainMap. As everybody knows, BrainMap is co-sponsored by the P41's funded Center for Mesoscale, housed at Martinos Center. So our speaker today really needs no introduction, but we still have to follow the form. So it's my great pleasure to introduce our speaker today, Dr. Anastasia Yendiki, who's an associate professor in radiology at Harvard Medical School and an associate investigator at MGH, Martinos Center, as you all know. She received her PhD in electrical engineering systems from the University of Michigan at Ann Arbor, where she worked on inverse problems in tomographic image reconstruction. She then came to the Martinos Imaging Center to learn about fMRI, but saw a tractography image on her first day and changed her mind. Her current interests are in obtaining accurate models of axonal projections from ex vivo microscopy, developing methods that can take advantage of these models to infer connectional anatomy from in vivo diffusion MRI, and one day getting good at flamenco guitar. And I think we're going to have a are we going to have a demonstration? All right, Anastasia, the stage is yours. Thank you so much for coming here today.
Thanks for the invitation. So my invitation email said, we realize you have never given a BrainMap seminar. And I was like, you just haven't gone far back into the prehistory of the Martinos Center. So I actually dug up, this is on the internet. My last BrainMap seminar was 10 years ago, February 12th, 2014. Same time, same place, same room. This room looked exactly the same. Some things never change. And then I had to actually go even further back into my own archives for, things that have been expunged from the internet. There was another BrainMap I gave in 2011. So the previous one was about that fun study on the effects of head motion in diffusion MRI that we had done with Nancy Kanwisher. And the 2011 one was when we first came up with Dracula, the first public release of Dracula. So that was also a BrainMap seminar. And I'm pretty sure there was one even further back, but thankfully, even my own archives don't go that far back. So even my bio, it was very similar. It was talking about how, what you just heard, how I came from an image reconstruction background, knowing nothing about the brain. I came here because I heard this is the place to learn about fMRI. But then on my first day, I saw a tractography image and changed my mind. So we'll see what has happened since then. So you see that the previous brain maps I've given are very much, in the sort of Martinos tradition of tool making. But today I hope to also talk about when should we stop making tools about to answer the same questions and take a step back and figure out when we need to start asking new questions. So where does the information that we have about how different parts of the brain are connected come from? There are two main traditional tools that anatomists have at their disposal. One is gross dissection of human brains. This is a natural fixed human brain that gets frozen and thawed and frozen and thawed, so that that process makes it easier to try to peel off individual fiber bundles. It's low precision. It's hard to follow individual fibers from all the way from their origin to their termination point. It's low throughput because it takes a very long time. It's a very time-consuming process. But what it has going for it is that it is feasible in human brains. And the other traditional tool in anatomist disposal is tracer injection studies. These are done in non-human primates because the injection has to be done in vivo and then the animal sacrificed. So these have very high precision, unlike the gross dissection approach. With a tracer, we can really see individual fibers from the injection site where the tracer was injected into the brain, all the way to all the termination points that injection site is connected to. It is also low throughput because these are also very difficult studies to do, time-consuming and costly, and cannot be done with too many injection sites in the same brain. And also, they're not feasible directly in human subjects. But a lot of what we know about what parts of the brain are connected to each other and the information that a neurosurgeon would use when, if they have to insert a sharp object into your brain, the information that they would use to figure out what they should cut through and what they shouldn't cut through actually comes from these kinds of studies, these tracer injection studies for the most part. So obviously it was very exciting when diffusion MRI was first introduced because it allowed both high throughput. So, in just a few minutes, you can image all the connections in the brain. And of course, it was feasible not just in human brains, but also in vivo, in vivo human subjects. But of course, the big question there is, what is the precision? And that we're still working on figuring out to some extent. So we know that it's good in some cases, bad in some other cases. And a lot of what I'm going to show is our efforts to pinpoint exactly where things go wrong and hopefully figure out how to fix them. So the very early studies that compare diffusion MRI to the conventional ex vivo anatomical techniques showed that for the most part, diffusion tractography was able to reproduce some of the big highways of the brain, like the superior longitudinal fasciculus shown here, or the inferior longitudinal fasciculus shown at the bottom, as they had been demonstrated either with tracer injection studies in non-human primates, as seen on the top, or with gross dissections in human brains. However, to get these correct representations of the big highways of the brain, it was necessary to do a lot of manual labor to tease apart these correct connections from all the non-correct ones in the whole brain diffusion tractography data. And so this set off an effort to come up with protocols for labeling specific white matter pathways in diffusion tractography data. These protocols that are still very much in use today are called multi-ROI protocols because they define each pathway based on a handful of ROIs that this pathway either goes through or doesn't go through. And this was first done manually. So for every single subject, you had to go in and draw these ROIs and isolate the spaghettis from the whole brain tractography that go or don't go through these ROIs, depending on these rules. And then the first automated approaches for identifying these pathways without manual intervention were based on that multi-ROI protocol. But here, instead of drawing the ROIs manually in every individual, you have them in an atlas space and you register them from the atlas to the individual. So this relies on an assumption that the actual streamlines that connect the ROIs that you want are somewhere in there. They're hidden in this whole brain bad hair day. But if those streamlines have not been found by the whole brain tractography, there is no way, as correct as your ROIs may be, to actually reconstruct them with this approach. And so we came up with an alternative approach that I had discussed back in my 2011 BrainMap seminar that's called tracula, or tracks constrained by underlying anatomy. And here, instead of first running the tractography in the whole brain and then finding the bundles of interest second, using a priori ROIs, here we are including all the a priori anatomical information about the bundles of interest in the step that generates the tracks themselves. And so it's a Bayesian approach that reconstructs the posterior probability distribution of a specific white matter bundle. This is the anterior thalamic radiation, by trying to optimize the fit of the shape of this pathway, both to the diffusion orientations from the subject's own diffusion data and to some a priori anatomical information about which anatomical structures, both cortical and subcortical, this bundle goes through or next to in the left, right, anterior, posterior direction, etc., at every point along this pathway. And this a priori information comes from a bunch of training subjects where this bundle has been labeled manually. Now, since then, a lot happened in diffusion MRI, and in particular, the acquisition technologies that we have at our disposal for diffusion MRI scanning have known just an explosive growth and great advancements, to great extent thanks to the Human Connectome Project. So the Human Connectome Project first developed some novel technologies for diffusion MRI, including both hardware and sequences. So scanners like the connectome scanner with stronger gradients, allowing greater diffusion weighting with better SNR, as well as accelerated sequences, allowing us to do that while at the same time increasing both the spatial and the angular resolution. So since we came up with that first version of Dracula, the kinds of data that are widely available became much better. Thanks to the Human Connectome Project that after developing these technologies also deployed them for a large-scale data collection. And after that first step of the HCP, those technologies were also used by other consortia, including ABCD and now the UK Biobank, even further increasing the amount of high-quality diffusion data that are available out there. Of course, most of the clinical studies collect actually lower quality data than the HCP protocols. And so one question that we asked was, can we use high quality training data from HCP style protocols and train Dracula to reconstruct the same tracts automatically in lower quality data with shorter protocols that are more likely to be used in the clinic. And so in that first early iteration of Dracula before the Human Connectome Project, it could only reconstruct 18 tracts that covered about 18% of white matter. And in the newer version that was published in this neuroimage paper by Chiara Maffei in 2021 and released the same year in free software version 7.2, I believe. It covers 82% of white matter and includes 42 large white matter highways. And importantly, it's not just the number of tracts that increased and the coverage of the brain. It's the kind of training data that we used. Back in the old version, we had used conventional data that were available back then, lower B values, lower resolution, single shell. And this is an example of the superior longitudinal fasciculus as we were able to reconstruct it after careful manual dissection from one of those training subjects. In the new version, we used training data from the connectome scanner in Bay 8, the now deceased connectome 1 scanner. And this is, you can see a full dissection of the 3 branches of the superior longitudinal fasciculus that were possible with these very high quality data sets. So then we wanted to test whether it was possible to use these manually annotated white matter pathways in these very high quality data that were collected with a B maximum of 10,500 plus directions to train Dracula to reconstruct the same tracts automatically in much lower quality data with only a single B value of 1,000 and 64 or even down to 32 directions. And so these are the results presented in Chiara's paper from 2021 that made it to one of the last this was our last neuroimage cover. We didn't know back then what was going to happen then with our favorite journal. What Chiara's paper is able to show is that we can get a very high sensitivity up to 89% in these lower quality data sets with a low B value, single B value, and just a few directions when comparing these automatically reconstructed tracts to the manually labeled tracts in the high quality data sets. But while with Dracula, to get to that high sensitivity, we have to incur a moderate reconstruction error of 3.5 millimeters when we have 64 direction data, or slightly higher, 4.2 millimeters when we have 32 direction data. With the conventional multi-ROI approaches that I showed you earlier, that reconstruction error that we have to deal with when we want to get to that very high sensitivity is actually much higher. So we want to take this a step further. And by the way, you should stop me and ask questions. I'm gonna like probably. Oh, hi.
Any ways to extend this further using AI-based kind of training and applications? Like how far do you think we can push it?
Yeah. So Bruce is asking a great question. Can AI do even better? And how far we can push this? So the main limitation here is getting those training data sets. So these are the same training data that we're going to give to the AI. So if these are wrong, if these tracts are wrong, then the AI is not going to be able to correct them because it doesn't know more anatomy than us. So really, we need the training data, and that gets us to the later part of the talk that's going to be about getting the ground truth in the brain. So once we have the ground truth, then we can take advantage. Yeah, exactly. So unlike, a problem of classifying between cats and dogs, where you can go on Google and instantly download 10,000 examples of cats and 10,000 examples of dogs, we don't have good real examples of what is actually connected to what in the brain. And so this is what's actually keeping us from really taking advantage of this growth of AI type methods in the last few years. But so taking this one step further, the idea of using high quality training data to reconstruct tracts and lower quality data, the next step that we're taking is trying to get even higher quality reconstructions of white matter tracts ex vivo that we can use to train our algorithms to reconstruct the same bundles in vivo. And this is just a toy example from one bundle of the superior longitudinal fasciculus that was manually annotated in just two hemispheres that were scanned ex vivo at 0.75 millimeter resolution. These were then used to train trachea, extract the anatomical priors, so learn what are the neighboring structures that these tracts go through or next to based on these ex vivo data, and then automatically reconstruct the same bundle at much lower spatial resolution, 1.5 millimeter in vivo diffusion MRI. So this is a demonstration that we can take this idea of quality transfer from higher to lower quality data one step further and take advantage of the much longer scans than we can do on ex vivo brains. And so why do we want to go to those even higher spatial resolutions? Because we want to go beyond these large highways. So we talked about these, the named pathways, the superior longitudinal fasciculus, the inferior longitudinal fasciculus, the cingulum bundle, et cetera. All these things have been, to a great extent, the focus of automated reconstruction, automated tractography methods. But then there are some bundles that are very important, for example, for deep brain stimulation, part of the reward system, that are much smaller than that. And so this is an example in some work led by Chiara Maffei and presented at ISMRM a couple of years ago, showing some of the small subcortical bundles that she was interested in dissecting. So the big bundle, the big round bundle that you saw is the fornix, and it's just shown here as a reference. And you can see it well, both in the 1.5 millimeter resolution Human Connectome Project data with the ones with the 500 directions and the maximum B-value of 10,000. And here in the higher spatial resolution 0.76 millimeter resolution with slightly lower b-value up to 2500 with a G-slider sequence. But the smaller bundles that you see here, the stria medullaris, the mammillothalamic tract, the fasciculus retroflexus, and the mammulotegmental tract, you can only see them in the higher spatial resolution 0.76 millimeter data, and you cannot see them in the super high b-value, high angular resolution data from the Human Connectome project, simply because they are below the spatial resolution of those data. Unfortunately, this is just one subject that was scanned over 18 hours, I believe, and averaged over 2,000 plus directions to get high enough SNR at this high spatial resolution. So we're hoping to be able to use these lower quality data that we can reconstruct these kinds of small tracts that are very clinically relevant automatically without having to put each patient in the scanner for 18 hours. And this is the focus of a NARSAD Young Investigator grant that Chiara has just started. So how do we know that all these bundles are there in the first place? We said that this information comes to a great extent from anatomic tracer studies in non-human primates. So one project that we started in collaboration with Suzanne Haber now about 10 years ago as well was to try to compare the diffusion tractography and the anatomic tracer injections side by side in the same macaque brains, so that we can figure out not just how often we fail to see a specific connection, but when we fail to see it, where exactly in the brain do things go wrong? Because you really need the side-by-side comparison in the same brain so that you can compare voxel by voxel to really figure out where things went wrong. And so we started scanning some of the macaque brains that had received tracer injections in Suzanne's lab at the 4.7 Tesla small bore scanner downstairs, collecting very high spatial and angular resolution and high b-value data over an entire weekend per brain. And we used these data to do a systematic comparison of different tractography methods, different orientation, distribution function reconstruction methods, and different diffusion sampling schemes to see which of these aspects of the diffusion acquisition and analysis pipeline makes the most difference in terms of the anatomic accuracy of tractography when compared to the tracer injections in the same brain. And one of the biggest effects that we saw was the difference between deterministic and probabilistic tractography. So these are ROC curves, the green ones with probabilistic methods and the red ones with deterministic methods. And so the two axes here, as in any ROC curve, are the true positives, true positive rate and the false positive rate. So when we are operating in low false positive, low true positive operating points, which is actually where the default settings of tractography algorithms usually are, there is very little difference in the performance of probabilistic versus deterministic tractography. But as we relax these thresholds to be able to see more and more tracts that we are missing at these low, very stringent thresholds, then is when probabilistic tractography becomes much more accurate than deterministic. And this mirrors some other studies of the same era that have done the side-by-side comparison of modern deterministic and probabilistic approaches. So we then use these data and share them with the community as part of an open tractography challenge that we organized in 2 rounds, mostly during COVID. And here the teams optimized their diffusion tractography pipelines for 1 injection site. and then applied these optimized pipelines to another injection site. So we had a brain that we gave them to train their methods, to optimize their methods, so that the tractography becomes as accurate as possible with respect to the tracer injection. And then we asked them to apply that optimized pipeline to a different brain that had a different injection in a different brain area to see how generalizable that optimization would be. And so the outcomes of that were published in another paper led by Chiara in Neuroimaging 22 were recommended best practices for analyzing diffusion MRI data. Again, probabilistic tractography outperformed deterministic. And we also had some in terms of how the data has to be post-processed to optimize the accuracy of tractography. And importantly, we also identified common failure modes across all the different DMRI pipelines. So what are the parts of the brain where most of the modern methods go wrong consistently? And it turns out that some of those common failure modes involved fiber configurations other than crossing. So parts of the brain where fibers are branching, fanning, turning, doing things other than crossing. So it turns out that we have gotten very good with our diffusion methods at resolving crossing fibers. But we haven't paid as much attention to these other fiber configurations that, as it turns out, are quite common in the brain. And this is the importance of having the tracer data to tell us how common these fiber configurations are. And branching is particularly common. And we know this is going to be common because we know that one part of the brain is not connected to just one other part, it's connected to multiple other parts. And so to go from one to many, we do need branch points. And so these are very clearly seen in the tracer injection data, and they're present for, regardless of the specific injection area. So for pretty much all cortical areas, because they are connected to multiple other cortical or subcortical areas, you have to have the fibers branching. And these were some of the trouble areas where tractography did the wrong thing. Then another interesting thing that the tracer data show us, where the reality deviates from some of the assumptions that we make as tractographers, are that not all fiber bundles are equally bundled. So you see here fibers, so the bright orange things are the fibers that came from an injection site in the anterior frontal cortex. And these are the fibers that from that injection site went into the internal capsule. So here you see the fibers very tightly bundled. So they travel very close, they stay close to each other as they travel through the white matter. But when you look at the fibers from that same injection site in the anterior frontal cortex that travel in other bundles, for example, inside the corpus callosum or inside the uncinate fasciculus, they are actually less bundled. So in the corpus callosum, they do tend to stay in the same area of the corpus callosum, but there are gaps in between, indicating that in those gaps, there are fibers from other cortical areas. And in the uncinate fasciculus, there are gaps, and also the fibers themselves that came from the same injection site are spread out pretty much all over the uncinate fasciculus. And this is very important for one sort of area of research that's very active within diffusion tractography, that where the assumption is that we can differentiate between different fiber bundles based on their microstructure. But if different fiber bundles are actually all mixed up, it's going to be harder to do that. And so this is an interesting observation that turns out to be not a function of where you injected, so not a property of the cortical area of origin, but a property of where inside the white matter, which pathway the fibers are traveling in. And yes?
Saying that distinction doesn't have any kind of cortical distribution pattern to it. It's just such as random on the cortical surface, then whether things track together or not, which is a white matter doesn't in any way relate to what we know about cortical cell architecture.
So it's not random in the sense that the fibers from anywhere in the cortex that go into the internal capsule are going to stay very close and bond to each other. And so that's why the internal capsule has a nice topographic organization where, depending on where you are in the internal capsule, you can guess where you project to in the cortex. But if you look at other pathways like the uns and fasciculus, you cannot really make that kind of determination because everything is kind of mixed up.
So in that regard, it's region by region, specific how these patterns either do or don't.
Yeah. It's specific to the white matter pathway rather than the area that the fibers originated in. That's a great question, but I mean, I don't know if we have the data to answer that question, but yeah, obviously the why is a big mystery.
Yeah. A question from the online audience.
Yes.
I don't know if you want to put it for the end of the talk.
It's okay.
Here's the question. It says, can you discuss the current and best possible theoretical resolution for crossing fibers? I guess you talked a little bit about that. But...
Yeah, so what we found is that at the resolutions that we're imaging, we've actually gotten pretty good at resolving crossing fibers. So the orientation distribution function reconstruction methods that we have are actually pretty good at that. So the bottleneck right now is not the crossing fibers from what we've seen in our work, is all the other kinds of fibers. Yeah, and one last thing, as an example of something that we see in the tracer data that kind of contradicts how we tend to think about things as structographers, is we commonly talk about superficial fibers as U fibers because we think of them as connecting one gyrus to the gyrus next to it. But actually, we see that superficial fibers have all sorts of shapes and may connect a gyrus to a sulcus or anything else in between. And so U fibers are actually a misnomer. And so we tend, as structographers, to kind of clean up our superficial fibers to only keep the nice U-shape ones. But actually, when we do that, we are getting rid of a lot of real superficial connections. And another line of work where we compared ground truth now in human brains where tracer injections are not possible to diffusion orientation distributions was led by Robert Jones a few years ago, where we compared diffusion MRI scans collected with different diffusion sampling schemes, including DSI, multi-shell, and single shell, and different downsamples to different spatial resolutions to polarization-sensitive optical coherence tomography, a method I'll show you a little bit more about that's led by Hui Wang here at the center, that gives direct measurements of axonal orientations. So the work by Robert compared these ground truth orientations from optical imaging to diffusion orientations in the same samples, and compared how these different aspects of how the data are collected and different ODF reconstruction methods impact on the accuracy of the diffusion orientations with respect to the ground truth optical imaging. So what he found was that the biggest effect was that of spatial resolution, much bigger than that of the orientation, the ODF reconstruction method or the Q-space sampling scheme, whether it was DSI, multi-shell, et cetera. And the biggest difference, the biggest jump in accuracy was when we went from a spatial resolution of 2 millimeters to 1 millimeter. And that shows us, again, the importance of trying to go to higher spatial resolutions and not neglecting the spatial resolution, in favor of the higher B values or angular resolution. Of course, the high B values are going to be very important for microstructural modeling, but for tractography, at least with the methods, the ODF reconstruction methods that we have now, it seems that they don't help as much as the higher spatial resolution. And again, this can change. may come up with other orientation reconstruction methods that can take advantage of these high B values. Yes, question.
Yeah, we have a question.
Yes.
From Hank Golding. He says, in resolving branching fibers, what are the roles of symmetric versus asymmetric ODFs?
Yeah, that's an excellent question. And indeed, asymmetric ODFs are one approach that has been proposed in the literature for dealing with these kinds of non-crossing fiber configurations. There has been some published work on this, but I think that it's something that we need to look into further and do more rigorous validation of these approaches. So in this approach, the broad idea is that you reconstruct the conventional ODFs at every voxel, and then you combine these ODFs within a neighborhood of voxels to try to figure out if you have branching or crossing or other kinds of things. But yeah, I think that's a very promising line of research. If you're working on that, keep working on it. Okay, so we talked about that. And going further than simply, optimizing the existing methods that we have, and, related to the topic that I posed in the beginning of when do we stop developing tools and take a step back and figure out if we should start asking different questions to develop tools for. This is work led by Chiara that she'll be presenting at OHBM this year, using the ex vivo data to sort of rethink some of these big highways. So we think of the bundles like the superior longitudinal fasciculus as one monolithic structure that goes all the way from parietal to frontal. And there is some controversy in terms of does it go, how rostral does it go in the frontal cortex? And there are contradicting findings in the literature. So we started working on this all the way back in my first R01, and I'm very happy to see this work that has been done thanks to contributions by many people now come together. So what Chiara is able to show is that some of these pathways that we think as a large end-to-end connection are actually made up of shorter relay fibers that do not go all the way from one end to the other. And so some of these things that we think of as structographers as one pathway may actually be composed by multiple shorter connections that use this pathway as sort of a highway to merge on and off of, rather than just follow it all the way. And this is something that she was able to show both with ex vivo diffusion MRI at very high resolution, 250 micron in small blocks that we scan on our 9.4 Tesla, and also with polarization sensitive OCT at even higher resolutions collected at 3 micron. And I mostly talked about white matter connections, but another very promising avenue for these more advanced diffusion reconstructions is microstructural biomarkers, and particularly for the gray matter, an area where the conventional diffusion MRI models aren't really informative. And this is work led by Ting Gong on using soma and neurite density imaging in non-human primate brains that were scanned on our 4.7 Tesla scanner with multiple B values and multiple echo times, and showing that the soma density and neurite density map that she extracts with this type of model match really well the contrast seen in Nissl stain and myelin stain, respectively, showing that the model is able to disentangle in the gray matter the contribution from soma versus neurites. And she has applied this type of imaging to a set of brains, developmental brains, going from 3-week all the way to 2 adults, A1 and A2 here, and looking at these neurite and soma density and soma size measures in different subcortical structures that you see here on the right, She is able to see both the developmental effects as you go from 3 weeks to adulthood, as well as differences between the different in the neuron and some density between the different subcortical structures. And these developmental effects are actually not seen with the conventional DTI-based biomarkers of FA and mean diffusivity, or with NODI, orientation distribution, and neurite density indices. So another very promising line of work for developing biomarkers that take advantage of these high B-value images that we can collect now in human subjects, hopefully also on the connectome too. And I will end, I have 15 minutes, so I'll just go over this very broadly, talking about a new effort that builds on all of these, little pieces that we have been doing over the last decade plus. And this is a project called the Large-Scale Imaging of Neural Circuits, or LINC, that is funded by the Brain Initiative Connects program. Connects is a $150 million total investment by the NIH to produce wiring diagrams of the brain in different mammalian species at unprecedented resolutions. And it has funded 11 total projects so far, of which 5 are UM1 comprehensive centers. And one of those 5 centers is ours, the LINC project. It focuses on macaque and human. It's a consortium led by us here at MGH with 7 collaborating institutions that will image cortical subcortical projections in both macaque and human brain down to the single axon scale over the next 5 years. So developing both the acquisition and the analysis methods to make this reality and get those training data that we really need to feed into the all-powerful AI. So we are linking across species, across different modalities, including diffusion MRI, light microscopy, and X-ray microscopy that I'll show you some cool images from, to ultimately link between cutting-edge ex vivo anatomy tools and in vivo therapeutic interventions, as I'll talk about when I talk about our use cases. The 3 MPIs of the project are myself, as well as Suzanne Haber and Elizabeth Hillman, with additional site PIs, Mike Fox, Satra Ghosh, Jeff Lichtman, Claire Walsh, Peter Lee, and Zhuhao Wu. The volume that we are targeting, those cortical-subcortical projections that I mentioned, are motivated by deep brain stimulation. And so it's a volume shown by this purple box here that contains the projections of the motor, premotor, and prefrontal cortex to the subthalamic nucleus, a very small structure that's shown here blown up in this very beautiful image by Barbara Hollander in this paper that has actually just gotten published in Nature Neuroscience, I believe. The different colors show the fibers that are connected to the areas that are associated with therapeutic effects of DBS, the areas of the subthalamic nucleus that are associated with therapeutic effects of DBS in these 4 different disorders. So dystonia, Tourette's syndrome, Parkinson's disease, and OCD. So which exact subzone of this very tiny nucleus that's just 1 centimeter across you stick the electrode in will determine the therapeutic effect for a specific disorder, as well as potential side effects. So it's important to know what fibers to activate to maximize the therapeutic effect and what fibers to avoid to minimize the side effects. And of course, this comes from a diffusion tractography image, and it does show us the overall organization, the overall topography of the large projections, but there's a lot that it doesn't show us. And in fact, most of what we know about the circuits implicated in these disorders and in DBS to treat these disorders come from those classical tracer studies, to the point that some of the atlases that are used to target DBS in real human subjects don't come from diffusion tractography data of those subjects, but come from anatomists like Suzanne here, having drawn wearing 3D goggles where these fibers go in a human template based on prior knowledge from her tracer injection studies in non-human primates. So clearly, we need a new tool set for imaging that's more reliable than in vivo DMRI is right now, and that can get us as close as possible to the precision of tracer injections in non-human primates, but that's applicable directly to human brains, so we can image these clinically relevant connections directly in the human brain and provide a resource that is going to be really invaluable to both basic and clinical neuroscientists that study how to improve deep brain stimulation and how to better model it so that to understand its how it works. So we have two competing demands here. We have both large fields of view that's needed to follow these fibers all the way from the cortex to the subcortex. And we need also ultra high resolution to be able to disentangle these fibers as they funnel through this very narrow passage inside the internal capsule. So if we try to satisfy both of those requirements at once, things will become intractable in terms of storage, data storage, and computation. And so we're going with this multi-scale, multimodal approach that includes three innovative microscopy techniques, as well as mesoscopic DMRI to provide the necessary link back to the clinical application. The first step is, of course, to do the tracer injections as the classical ground truth to compare all the innovative microscopy techniques that will be performed downstream in macaque brains. Excuse me. In the human, we're going to use some of those topographic organizations that we mentioned earlier, and our ability to be able to reconstruct these topographic organizations in the human brain will be sort of our validation method for whether what we're getting with the novel microscopy techniques is actually right. The first step in our imaging pipeline is, of course, diffusion MRI here at the Martinos Center. In the human brain, this will be done in the connectome 2. And this, of course, is a project that has been going on over the last few years, led by Susie Huang. And finally, the scanner was installed this past summer with great success. It's the highest gradient strength system in a human MRI system with a Gmax of 500 millitesla per meter and slew rate of 600. And it includes custom instrumentation for performing ex vivo imaging of whole ex vivo human brains, including this custom 64-channel coil. After the diffusion MRI is when we'll start slicing and dicing the brains to get at even higher spatial resolutions. And the first step here is the method that I mentioned earlier that we have previously applied in just small samples in the human brain. Now Hui is going to take this approach, the polarization sensitive optical coherence tomography, to entire hemispheres. So PSOCT gives us label-free, so no staining, direct measurements of 3D axonal orientations, and the plan is to do this at 600 micron resolution to really resolve microscopic fascicles. And an additional benefit of PSOCT, in addition to its high resolution, is that it images the tissue surface first before it is cut. And so because the imaging is done prior to cutting, there is no histological type slice-to-slice distortion. And so you can really stack the images from slice to slice without having to solve a complicated registration problem and get these nice volumes like what we see here. And for the macaque, this will be integrated with simultaneous light sheet microscopy led by Elizabeth Hillman to image the fluorescent tracers from Suzanne's injections directly in one go together with collecting the PSOCT data. And an additional advancement in the PSOCT technology that will be implemented by Hui and that was first implemented by Chao Lu, who was a postdoc here at the center up until a couple of years ago, is to collect the PSO-CT data with multiple light incidence angles. Each light incidence angle gives a single measurement of the in-plane orientation of the fibers in the plane that's perpendicular to the light incidence angle. So if you collect that with multiple light incidence angles, you can infer the through-plane orientation, as seen in this beautiful whole section, coronal section from a human hemisphere. Then the plan is to take these sections, clear them, and then perform immunolabeling with a variety of axonal markers, and then followed by light sheet microscopy to go to even higher resolution, a sub-micron resolution, to image individual axons. And this builds on work that Zhu Haogu and Elizabeth Hillman have been doing as part of their cell census network project with two key technologies, HubClear or human brain clearing, and HOLIS or human brain optimized light sheet that allows immunolabeling with a variety of markers and spectrally multiplex 3D volumetric imaging at super high resolutions, as you can see here. And last but not least, we have an additional innovative microscopy technique called HIP-CT, or hierarchical phase contrast tomography, that allows label 3 imaging at microscopic resolutions of intact organs. And this will be followed up with micro-CT. So the HIP-CT is led by Claire Walsh and Peter Lee at UCL. And this will be followed up as a validation with a more conventional osmium staining-based technique, micro-CT in small samples to validate this innovative HIP-CT contrast. So this is a video from a whole human brain that they imaged with HIP-CT. The technique is only feasible in one synchrotron facility in the entire world, the European Synchrotron Radiation Facility, shown here. And it takes advantage of faint tissue density contrasts to image with X-rays in intact brains, starting at resolutions of 20 or so micron in entire human brains, and going down to even higher resolutions of a few micron in smaller ROIs within that intact brain, allowing true multi-scale imaging down to the cellular level. And in smaller X-sized ROIs is where we plan to compare to micro-CT to really validate that we are getting myelinated axons with this novel HIP-CT technique. And this is a brain I was able to visit the synchrotron just a couple months ago, 1 month ago, actually. And this is the first brain that we have imaged for the project at 20 micron resolution, showing really amazing anatomical detail. And last but not least, we are going to take all these data and our computational team between Satra, Kabi, and Aaron at MIT, Yael, and myself will have our hands full trying to analyze the data, to perform cross-modal registration, to combine the different modalities, axon segmentation, as well as multi-scale tractography, combining all the information across modalities and scales. And to end, our two use cases. One is to use these data to map the cortico-STN circuits associated with therapeutic or side effects of deep brain stimulation to the STN. This is led by Mike and Andy at the Brigham. And last but not least, to use the data to investigate the mapping between the ground truth microstructural features of the axons and the diffusion MRI signals, particularly for extracting those biomarkers with these advanced diffusion microstructural modeling techniques. And this is, of course, led by Susie and Hongxi here. And I want to end by thanking the UM1 team. None of this would have been possible without their hard work and sacrifices. And thank you for your attention.